Midterm project.
Ariana.
Thomas.
Casey.
Our experiment originally simulated rattlesnake venom and how various variables affect enzyme reactivity, such as temperature, pH, and substrate concentration. Our experiment is based on varying levels of pH.
Our research question is, how does pH affect the activity of rattlesnake venom as an enzyme to deactivate its target? Background information includes the type of 4 types of molecules: carbohydrates, lipids, nucleic acids, and proteins. The monomer of carbohydrates are monosaccharides, such as glucose or fructose. The polymer is a polysaccharide, like starch. Carbs function as an energy source in the form of ATP and tag lipids and proteins for cellular communication.
Lipids. The monomers of lipids are glycerol and fatty acids. The polymers are triglycerides or phospholipids. Lipids function as a structural component of cell membranes and store energy.
Nucleic acids. The monomer of carbohydrates are nucleotides. The polymers are DNA and RNA. Proteins. The monomer of proteins are amino acids, while the polymers are polypeptides. Proteins provide intracellular structural support and act as enzymes to catalyze chemical reactions.
Enzymes are proteins that act as catalysts to speed up chemical reactions.
Proteins fold in a specific way because the sequence of amino acids in the primary structure dictates how they interact with each other through hydrophobic and hydrophilic units.
Primary is an amino acid sequence of the polypeptide chain. Secondary are interactions from A helices and B-plated sheets. Tertiary are more compact structures forming protein domains, distinct structural units that fold independently. Quaternary are composed of multiple tertiary subunits. Our hypothesis was that if the pH is altered by the addition of an acidic or basic solution out of the neutral range, the enzyme will denature and the reaction rate will decrease significantly.
Our independent variable is the change in pH by the addition of HCl or NaOH.
The dependent variable is the time it takes for the reaction to go to completion.
Our control group was the test tube that did not receive any acid or base solution.
The controlled variables include concentration of enzyme in solution, the temperature of the water bath, the volume of enzyme solution, the volume of substrate, and the volume of Benedict solution. Conclusion. Our hypothesis was ultimately rejected. Our results indicated that the acidic solutions had a faster reaction rate by about 35 to 45 seconds. There are 2 main classes of bacteria, gram-positive and gram-negative. Gram-positive bacteria have a thick peptidoglycan layer and no outer lipid membrane. Gram-negative bacteria have a thin peptidoglycan layer and an outer lipid membrane. During gram staining, gram-positive bacteria retain a purple stain due to the thick layer, while gram-negative bacteria lose the stain, revealing the thin peptidoglycan layer.
Antibiotics, such as penicillin, work to inhibit the enzymes involved in the synthesis of the peptidoglycan layer. This leads to weakened cell walls and ultimately results in cell lysis due to osmotic pressure.
Antibiotic resistance in bacteria. Genetic mutations. Random mutations in bacterial DNA can lead to changes that confer resistance. This can enable bacteria to survive despite the presence of antibiotics.
Horizontal gene transfer includes when bacteria can exchange genetic material, acquiring resistant genes from other bacteria, which allows resistant traits to spread rapidly across different bacterial species. Biochemical mechanisms occur when some bacteria develop mechanisms to pump out antibiotics before they can take effect, or they produce enzymes that break down the antibiotics, neutralizing their impact.
Our medical intervention. Rednicocillin is a tetracycline antibiotic. It enters gram-negative bacterial cells through specialized protein channels called porins in the outer membrane. Our drug is an altered version of the existing drug, doxycycline. This drug is lipophilic, meaning it dissolves in nonpolar solvents. Rednicocillin, however, contains an extra OH group, making it hydrophilic. As it now dissolves in H2O, it can enter the porins and inhibit ribosomal function.
Once inside the gram-negative bacteria, it binds to the large subunit of the bacterial ribosome. It binds to the peptidyltransferase center on the large subunit, directly preventing the formation of peptide bonds between amino acids. The PT center of the ribosome catalyzes the formation of peptide bonds during protein synthesis by aligning 2 tRNAs. Rednicicillin binds to the amino acyl PT and inhibits the induced fit mechanism that positions the amino acyl tRNAs together.
This ultimately prevents formation of the polypeptide chains required to create proteins and enzymes within the bacteria, resulting in cell death.